Brown University Data Science Initiative Symposium on Decoding the Pandemic. My name is Joe Hogan, and I'm very pleased to be able to introduce Natalie Dean. So Natalie, I hope I won't embarrass you too much by saying if anyone has watched really any of the major networks or has been reading the Washington Post, you've been introduced to Natalie on TV or through her opinion pieces in the Post. Natalie has really been out front, I would say, among biostatisticians and really among data scientists with sort of doing exactly what the title of this seminar is decoding pandemic data. She's really helped us weigh in on everything from bias sampling to uncertainty in models, to how to design seroprevalence surveys, and a lot of things in between. And Natalie's been a confident and authoritative voice on all things COVID data related. And we're really, really pleased that she's taken the time out of her schedule to spend this hour with us. So Natalie's, in addition to her work as a collaborator with a modeling group at Northeastern and her scientific communications efforts that she's been undertaking during COVID, Natalie is also part of a group that has laid out strategy for conducting randomized trials during a pandemic, and then more specifically, vaccine trials. And I think vaccine is on the minds of everyone because we see that as sort of the gateway to whatever the new normal might look like. So we're all paying very close attention to what's happening in the world of vaccines, and it's really terrific to have someone sort of maybe guide us through and help us understand how that process might unfold. So without any further introduction parts, I guess I should say one thing I didn't is that Natalie is an assistant professor of biophysics at University of Florida. So that's her affiliation. So thanks again, Natalie. I'll turn the floor over to you so you can share your screen. We'll go about 30 minutes, and then we'll open the platform for Q&A. I guess I should say too, we have a lot of participants today. I expect more will join. If you do have a question along the way, you can post it to the chat. And if there are a lot of questions or a lot of raised hands at the end of Natalie's presentation, I'll help sort through the questions on chat and call on individuals. So if you do post your question in the chat, I'll either, maybe you could just let me know if you want me to read it out loud or you'd like me to call on you. Thanks again. And Natalie, I'll turn it over to you.
Great, thanks so much for having me. So I look forward to chatting with everyone. I'll talk, my presentation's about vaccines, but at the end, I'm happy to chat more generally about whatever people want to talk about. So let me share my screen. Okay, can see that?
Mm-hmm.
Okay, great. So I'm an assistant professor at the Department of Biostatistics. Today I'm going to be talking about COVID vaccine evaluation. So first, a little background, some basics about vaccines. So there's a very typical regulatory pathway for vaccines. It starts with the collection of preclinical data, that's animal data. So, you know, we start developing vaccines at the bench in the lab, and then we test them in an animal that is a validated animal model that replicates the symptoms of the disease, so we can tell whether there's evidence of safety and anti-disease activity. So one challenge here is that requires a validated animal model. For a new disease, we haven't had that, so that work's been ongoing. But then the vaccine moves into clinical trials. So there are 3 basic phases. The phase 1 trials are the first in human trials. They're fairly small. The focus is on safety. Individuals are very closely followed for any adverse events. And one of the goals is to establish dosing. We also start to look at evidence of immunogenicity. Phase 2 trials are slightly bigger, and there's a larger focus on immunogenicity. So we're looking at the evidence of an antibody response in individuals, and whether we think that might indicate protection against either infection or disease in the future. But the way that we actually establish whether a vaccine works as intended So to establish the efficacy are through these large phase 3 trials. So these are our field trials. These can include thousands, tens of thousands of participants. Typically, individuals are randomized to either vaccine or some placebo, and we follow individuals to look for a disease-related primary outcome. So typically, we're looking, does the vaccine actually prevent disease, symptomatic disease, laboratory confirmed symptomatic disease, not just infection. And so we're seeing like people who are actually out and about and naturally exposed through in their communities, are they actually being protected? So this pathway usually takes a while. And so, one consideration is how, in the context of a pandemic, how can we accelerate this process? And so there's a nice perspective piece in the New England Journal from the group at CEPI, which is the Coalition for Epidemic Preparedness Innovations, that focuses a lot on investment in vaccines. And what they discuss, the outbreak paradigm involves not skipping steps, but overlapping steps as much as possible. So usually when we move between the phases of trials, you finish your phase 1 trial, you analyze the results, and then you make a go, no-go decision about moving to the next phase. But here in the outbreak paradigm, as much as possible, we overlap steps. So there's more investment up front, so it's a riskier investment because you may decide that you are not going to advance forward based on earlier data, even though you've already invested resources into starting that next step. So there's more financial investment, but that can accelerate the process. Another thing done in parallel, well, there's two things. So one is the collection of animal data. So some of these vaccines have moved into human safety studies without as much animal data, and that usually is because we have some evidence that the product is safe for other reasons, like that platform has been used for other diseases. So we have some good data that it's safe in humans. So those were allowed to move forward while animal studies are being done in parallel. And then also looking at large-scale manufacturing. So ramping up manufacturing in parallel, even before we know the results from an efficacy trial, whether the product is effective. So there are over 100 vaccine candidates currently in development. So that's another big part, a parallelization of effort. So there are a lot of efforts. We're not putting all of our eggs in one basket. There are a lot of different vaccines being pursued, and they use a range of vaccine platforms. So this is just a very general table describing the attributes of these different types of vaccines. And so some of the ones that have been fastest out of the gate have been these RNA or DNA vaccines, where you basically can plug in just the genetic sequence of the pathogen, the virus, and use that to develop a vaccine. So you don't need to isolate the virus or grow the virus. You're just sort of plugging it in. And these platforms have advantages for pandemic situations because they're very fast to develop. But one of the challenges is that they haven't been licensed before. We haven't had any successful RNA or DNA vaccines so far, so it's a little bit more uncertain how they'll work. Also, they tend to not be very reactogenic, eliciting as strong of an immune response, so they may require multiple doses. So basically, we're hopeful that these work, but we're not putting all of our eggs in that basket, and still pursuing some of the other types of platforms that may not be as fast to develop, but we know can work well, and also may be easier to manufacture as well. So so the WHO has a pretty updated, what do they call it? Sort of state of where everything is right now. Draft landscape. And so they update that regularly. This is from a week ago, and it indicates that there are 8 vaccines that are currently in clinical evaluation, meaning they're in either phase 1 or 2 trials. So this is just a summary, so you can see they use the different types of platforms. RNA, that's the Moderna NIAID one that we hear a lot about. And then they're in the University of Oxford, and they're in some mix of phase 1 or 2 trials. Sometimes what we're seeing are that these phase 1-2 trials are combined. Basically, you're just enrolling a lot of people, bigger than what a normal phase 1 trial would be. And you're looking at safety and immunogenicity in everyone. And right, and like I said, some of these have been able to move forward because these platforms have been used for other, you've used the same platform for other non-coronavirus candidates, or coronavirus also including SARS and MERS though. Okay, and then like I said, in parallel, we're also seeing, so we're seeing emerging data from those phase 1 trials, although still very early. And in the same time, we're also seeing data from the animal model, the animal studies, because those, like I said, are being done in parallel. Okay, so what we should expect to see, are these emerging results both in these small groups of people looking at safety and immune response, and then animal studies as well. Okay, so that's sort of where we are right now. So I'm going to focus on trial design, which is where my research focuses. So since 2016, I've been working with the World Health Organization's R&D blueprint for action to prevent epidemics. This was formed after the 2014-2015 West African Ebola epidemic. And there are several different work streams. The goal, the overarching goal is that there's more that we can do before epidemics to make sure we're ready to do R&D, to develop vaccines, therapeutics, diagnostics. So what can we have in place before a pandemic starts in order so that we can be most efficient and ready when something happens? So there are different work streams related to getting the funding coordinated. There's a work stream that relates to getting the labs, organized. And then there's a work stream that relates to identifying prioritized pathogens. So these are pathogens that an expert committee thought was most likely to cause a public health emergency, and for which we lack any effective vaccines or therapeutics. So some of these, you may have heard of, some of these you probably haven't, or maybe haven't, I don't know. But Ebola, and then it included MERS and SARS, so Middle East Respiratory Syndrome and the original SARS, so both severe coronaviruses. And then it also includes disease X. So this was pushed by my mentor, Ira Longini, and to include this in the list. And basically it means some as yet unidentified threat that can cause a public health emergency. And so that's exactly what COVID is something that we didn't know could be a problem, so we weren't something emergent. So I've been working on a work stream related to clinical trial design, so developing norms and standards for how we evaluate vaccines in the context of an epidemic. So we've made very general recommendations about how to design vaccine efficacy trials. So this is one paper where we kind of go through how should you define the outcome, and what populations should you focus on, and sort of general principles. But then I've also been approaching this from a research angle. So in 2018, I got an R01 called the Design and Analysis of Vaccine Trials for Emerging Infectious Disease Threats. And we focused on devising innovative vaccine trial and study designs that are tailored to outbreaks. Want to provide guidance on study design and analysis, examine the statistical properties of our proposed strategies, and then we use modeling to evaluate their robustness. So our team at the University of Florida includes me, Ira Longini, Yang, Sam Wu. We have Betts Halloran at the University of Washington, and then Alex Vespagnani, Marco Aielli at Northeastern University, who do this very sophisticated computational modeling. I'll show you some examples. So we've approached this problem from a few different ways. One is to think about the specific diseases and how would you design vaccine trials for these specific diseases, but also to identify just some common challenges that persist with all of these outbreak pathogens. So what are going to be the common challenges for vaccine trials, and can we identify common solutions? So some of the biggest challenges that in outbreaks, incidence is very unpredictable. And it tends to be very localized, but changes a lot over time. It's very hard to know where an outbreak is going to take off and when. And then when you look across a large population, incidence can be fairly low. This is especially true, with like Ebola, for example. It was a huge outbreak that occurred in West Africa, but it was like 0.3% of the whole population or some, it didn't affect most people. So on average, the incidence is very low. Outbreaks are a variable duration. They can start and stop. So people, different regions will get outbreaks under control. So incidence is on the decline in New York City, and as other areas hopefully employ better containment strategies, then incidence will decline there as well. So all of these contribute to challenges of low power. It can be very hard to collect enough events to establish efficacy, to establish whether the vaccine is actually working. Then there's, in the outbreak setting, we have poor understanding of the disease or the vaccine. We don't have the luxury of time to figure out all of the, do we think that the vaccine might wane over time? Do we know how, the features of the disease? So just in general, we're learning as we go. So there's just a lot more uncertainty when we plan. And then, typically outbreaks occur in resource-limited settings. And as we move forward, we're going to see more, I expect to see more outbreaks in resource-limited settings. And so when we think about conducting these trials, we may, move into the areas that are sort of hotspots. And these may have more limited infrastructure or experience doing clinical research. So feasibility is a very important principle. Okay, so I'm going to talk about some basic solutions that we've been proposing, just sort of overarching principles. So the first is flexibility. So before working with the blueprint, the way I got involved in that work is that I was part of a team that did a phase 3 Ebola vaccine trial that ended up being successful. So the basic idea, it used this innovative design called a ring vaccination design. And the basic idea is that every time a new Ebola case is identified, you go and you use contact tracing to identify their contacts. So these are their neighbors or their family members, and then the contacts of contacts. So the second layer. So who are those neighbors in touch with? And this forms a ring, and then you can randomize that ring. So we did it as a cluster randomized, but it could be individually randomized within rings, people to either immediate vaccination or delayed vaccination. You could use a placebo, but there's a lot of debate about what was considered acceptable at that time. So, so one of the things that was so innovative about this is that it was really a mobile trial. So, instead of people coming to a hospital or some site to get vaccinated, the trial went to them. And so it was, the PI called it a trial in a box. So you put all your equipment in these big tubs, and then you go out to these communities, and that's how you do your consent and enrollment. And then you go out and you're doing follow-up. But if you're following up every 2 weeks or something, then you're going out and you're following everyone up at that 2 weeks. So it ended up having a really nice structure. And it was particularly successful because this is the epidemic curve in Guinea in the West African Ebola epidemic. And the decision to conduct the trial occurred around the peak of the epidemic. But what happened is it takes time to get a trial set up. And by the time the trial started, the outbreak had started to wane. But because it was so highly localized and focused, we were able to get the data we needed to establish efficacy. And so randomization was stopped here after an interim analysis demonstrated efficacy of the vaccine. So it was this highly focused strategy that was able to catch this tail. And while other trials that were conducted during that same time They faced challenges because there were no more, in the places that they had set up, there was no more Ebola. So they were able to collect a lot of safety data, but no efficacy data because there were no infections observed in anyone in the trial. So how, so these flexible strategies are one approach. Another is, can we be very smart about the study sites that participate? And so ideally where we want to place the trial is in sites that have the highest incidence of disease. And that, that's also like the incidence that you're expecting to see over the next few months. So, so you can make decisions based on surveillance. So if you have good surveillance systems, you can look and see where are things starting to take off and try and place your trial sites there. But you can also use things like spatially resolved models to formalize your decision process. And so this is something that was discussed for Zika. So the group that I work with at Northeastern had this a very highly spatially resolved model for Zika transmission. It used temperature, airline data to model movement between populations. So it used a lot of different, information about population structure, and so able to make, a pretty very spatially resolved model about where we expected transmission to occur. And this was used. This is unpublished stuff, but it was used in discussions with NIAID to think about setting up a vaccine trial. So where can you identify the potential hotspots? So I'd like to see something similar occur for COVID-19, and this is something I'm working on right now. This figure has nothing to do with COVID, but the basic idea is that if you one strategy we can pursue is using ensemble modeling. So any particular model may be unreliable, but if we can use multiple models together that can each make projections about where, what they think incidence will be over, say, the next few months in a set of sites, then we can combine those models using, I like this figure, this is a stacked prediction. So this is basically a weighted average of these densities. So you're combining these densities, and then you can get a sense of what the models say is kind of the full range of possibilities. Maybe, there's some chance that things will really take off, or maybe, there's some chance that we don't expect any transmission in an area. So one thing I'm thinking about right now is the use of this ensemble modeling. How can we create something that would inform the people who are trying to identify these trial sites? So this is something that my postdoc made, and so can we create like a Shiny app or some sort of tool that allows people to sort through, if we had these combined model projections and then we had some other data, can we create some tool where people can kind of click and say, okay, well, maybe these are the places we're prioritizing. So there's a lot of other, you wouldn't just pick the top ones. There are a lot of other political reasons, operational reasons why you would pick a certain set, but trying to provide sort of a nice, useful tool. So that's something we're thinking about right now. Okay, so flexibility is a key feature, and then another key feature is collaboration. So this is a slide I made a while ago, so these numbers are old, but pretty quickly a lot of clinical trials were launched. This is focusing on treatment trials, but it's the same basic idea. There were over, by mid-February, there have been over 80 clinical trials launched to test coronavirus treatments. So most of these being in China, that's where the epidemic was at that time. But what happened to a lot of those trials in China? Well, so this is the number of new cases per day in China over time. It's on a log scale. But we know that China's done a good job of keeping their numbers very low, whereas everywhere else the numbers it's taken off in a lot of other places. So so these trials in China, a lot of them had to stop early because they no longer could accrue people who needed treatment because no one was sick and needed hospitalization. So some of those trials, the results are coming out now, and they're largely underpowered because the trials had to stop early. But if we had some structure where that trial could, instead of having to stop, could pick up somewhere else, then that would allow those trials to continue. So our basic recommendation, so this came out about a month ago, but it had been in the works for a while, is that you need a framework for conducting this type of research under something that like a master or core protocol. So we want some sort of setup where different, instead of places starting new trials, they can contribute data to the same trial. So what we're really trying to avoid is the publication, we're really trying to avoid these underpowered results that are very confusing to interpret, that sometimes get published, but then no one quite knows what to make of them. So we want to avoid the publication of inconclusive results from underpowered trials that can confuse decision making. And then if they look promising, these underpowered results can sometimes jeopardize our ability to move forward with randomized trials in the future. So we can end up in this perpetual state of uncertainty where we don't know if something ever worked. In the paper, we talk about how one big trial is better than many smaller trials, and that by working together, we can rapidly accrue numbers, and that's how we're best equipped to achieve answers quickly. And you need to have a structure in place kind of from the start that allows you to expand to different locations and even if necessary, extend the protocol into subsequent outbreaks. That's not going to be an issue with COVID, I don't imagine, but for other diseases we've dealt with like Ebola or MERS, the idea is that you may not be able to answer a scientific question within one outbreak, and you might need to pause it between outbreaks and then pick back up again. So with that in mind, the WHO launched a global mega trial of the 4 most promising coronavirus treatments. So the Solidarity, the multi-country Solidarity treatment trial. And so that uses a core protocol, and there are a lot of countries participating from all across the world. And so randomization is ongoing now. And places continue to be added as they get regulatory approval. Okay, so I'm just about done. So the thing that I have been working on lately is the Solidarity vaccine trial, which is closely related to the Solidarity treatment trial. So I'm part of a working group that's been developing a protocol or strategy for a multi-country phase 3 vaccine trial. And so, you know, we're putting, I'm not going to go into a lot of the details about the specifics of the endpoints and things like that, although that's what we're laying out right now in the document. But the basic idea is that it's a very large trial, multi-country trial, and that will allow us to sort of fastly or to quickly enroll and vaccinate participants in lots of different areas. So maybe using this mobile structure as much as possible. And we want things to be adaptive. So it's not going to be just about one vaccine. It's a standing trial structure that vaccines can enter into. And so we can evaluate multiple vaccines at once. Okay, so that, I think, all right. So that's what I have. So I went a little quick at the end, but I am happy to answer questions now.
Natalie, thanks very much for that. This is really quite a tour. It's funny, I was jotting down questions as you were going, and then every time I jotted down a question, you answered it like in the next slide. So I'll ask the audience to use the raise hand function. I was going to explain how to use it, but I don't know if I know myself. I believe if you go to the participants tab, then you'll find it in there somewhere. You can also send a question on the chat if you would like me to ask the question for you. So let me just start with one or two to get the conversation going. I was really, I was really intrigued by the sort of interface between design and then modeling, and how that you kind of go back and forth there, because the first talk we had was by Betsy Ogburn. She talked a lot about sort of collaboration in clinical trials, and a lot about clinical trial design and combining evidence. And then Nick Jewell talked about modeling and the uses and misuses of models. And now, we see them both together in the sense that you are talking about, well, how do we target where to roll out the vaccines and things like that. So, I was, I guess this is the question you sort of answered. You might want to say a little bit more about this, that a lot of the models that we've seen have come in for criticism for being inaccurate in their projections. And personally, I just, as someone who's just done a lot of data analysis in the past, I think that there's a huge burden on some of the modelers, because it's really, really hard. As they say, prediction is really hard, especially when it's about the future. And when you use these models, like, how, is there sort of a feedback loop that tells you, are we getting it right as far as where the breakouts are coming, and how much do you really lean on these? I'm just kind of interested in that interplay.
Yeah, absolutely. I mean, I think we should be very skeptical of any particular model's ability to predict the future. It's just enormously difficult. And we've seen that play out over and over. And I think the way that I think about models here is just a way to formalize or synthesize the available data. And I mean, we can look, there's very informal things we can do, like looking at the current incidents, or we can look kind of back at what's happened before. But the model at least is sort of it's just a formal way to say, what do you think is going to happen? And of course, there's a wide range of uncertainty, and so one of the roles of the stacking, for example, is that you can use the models not just to get a point estimate, but also to get a range of uncertainty. And you use different models to even get more uncertainty, because any individual model, their uncertainty is surely an underestimate of the uncertainty, because it's like statistical uncertainty, but then there's also this uncertainty in just if you're missing some important feature of actually what's going on. So just, but still at the end of the day, the core of the strategy is that you don't just pick one place, you pick a lot of places, because there's so much randomness in where things really take off, that the idea is that if you enroll enough places simultaneously, then even if it doesn't take off in some of them, by chance you expect it to take off in one of them. And you can quantify that more, and that's something that my postdoc is working on. But for now, it's kind of don't rely too much on the models to make predictions.
Right, but yeah, like in the way I guess you have the same sort of quasi-skeptical view, using them to help target where to roll out the different components of the vaccine trial.
Yeah, I think it's just a way to formalize your intuition, but at the end of the day, you need to what we want is just to sort of have as many sites participating as are able to. And so one of the things about the core protocol is you're making the barrier for entry low. You're making the procedures simple, the approvals as simple as possible. Like this concept of those large, simple trials almost. You're keeping things very simple so that it's easier for places to participate.
Right.
Yeah.
Okay, we have a question from Jim Padbury. Jim, I took the mute off. I don't know if you want to ask this question on your own, having to do with merging results from different vaccines.
I don't hear anything.
Well,
Okay.
I'll ask the question then. So this comes from Dr. Jim Padbury, who's here at Brown. He says, in a solidarity trial, a term I'm not familiar with, Natalie, but maybe you are, how can you merge results from different vaccines?
So within the context of a single trial,
Well, let's see. We're trying to get Jim unmuted here. Sorry for the
That's okay.
technical glitch here.
Yeah, because there's two, I mean, there's both within the context of a trial, there are going to be multiple vaccines. So they're entered in as different arms against with a shared placebo arm. And you know, there are discussions now about kind of what happens if one of them is shown to be efficacious, and then how does that, you know, but at the end of the day, the goal is to find as many different vaccines that work as possible. So we're not trying to find the one best. We're trying to find as many as possible. So we're not going to drop drop other arms very early. But if that's the question,
Yeah.
I'm not sure.
So Jim's having trouble with his microphone, but he says, so they are indeed analyzed independently despite the shared control.
Yeah. I mean, I think you have to. If people are going to participate, they, you know, yes, they kind of need to be. You wouldn't want to. We want different groups to participate as much as possible. So otherwise, they're all going to do separate trials on their own. And I think what a lot of manufacturers are going to do is do both. They're going to participate in this large trial, and then they're going to participate and maybe do their own smaller trial. But the point is that we have this large network of places. So yeah, there's no plan for a multiple comparisons adjustment.
I see. Okay. I'm going to go to Sam Fulcomer, who's also at Brown. And Sam, I believe we've unmuted your microphone, so go ahead.
Great. Well, first of all, thanks very much for the talk. I was just wondering about the controversy over challenge trials. And how you see that playing out.
Yeah, absolutely. So human challenge trials, for those who don't know, are trials where you have voluntary participants, and some of them are vaccinated, some of them are not vaccinated, and then all of them are exposed to an inoculum. They're intentionally infected. And then that's a way to, in a well-controlled set, you can use really small numbers in that case to tell if something works, because with field trials, you need large numbers to get even just a few people to see a few infections. But here, you know, if everyone's infected, you can see. The challenges with them, and these are being pursued in parallel, so these are being discussed right now. The challenge is that it's going to be hard to get approval for that when you don't have a treatment for an effective treatment. So so there's just some ethical concerns there, of course. But if people give informed consent, you know, that if people can really give informed consent, then those might still be approved. But they're very slow to set up, and they're probably going to preferentially enroll just very young people who are going to be at the lowest risk of severe disease. And then that's not going to be sufficient information to license a product for older individuals. It may not translate to whether it actually works in older people. And so it's not going to provide all the information that regulators need to make decisions. And I'm not sure that it's also faster. I'm not convinced that it's faster, but because they take so long to set up, and because we're really barreling towards these phase 3 trials. So, and because there's so much circulation right now of COVID, that if you put a trial in the right place, I think you could accrue numbers pretty quickly.
Okay, thanks, Sam. We do have one sort of generic question I wanted to read out because it's probably something that's on folks' mind. Just the only name we have for this participant is Nisarg, who says, there's been criticism of Moderna's trials. What is your opinion? Now, I know that's sort of putting it generically, but maybe to put a bit of a finer point on it, you talk at some point about the about this effort being somehow collaborative, and that there's lots of small trials with low power, we may not learn as much as we'd like to. I mean, that relates a little bit to the shared controls issues as well. But do you have any comment or something to say about Moderna's efforts and where they fit into this larger spectrum of efforts?
Yeah, so Moderna has been furthest along. They were the first group, so this is the RNA vaccine. They were the first group to, first in human, to actually vaccinate people. And they're using this RNA product. Yeah, I mean, they're moving quickly. I think that's great. The results that were released were very premature. You know, it's just eight. It was like, I think 8 people or something. So clearly that's not I mean, I think large numbers of people have been included in this. I mean, I think over 100 at least. So, but I mean, I think the best way to interpret the results, I do believe that that product is probably safe. I mean, we have experience with that product from other non-coronavirus diseases. And so that was one of the reasons why it was allowed to move as quickly as it was without animal studies being completed, is because we had evidence that it's safe. But you know, but in terms of the immunogenicity and things like that, it's really unclear, you know, what was going on exactly and how to interpret the results. I think it's just far too early.
So is there going to be, like, you know, again, you talk about this sort of, well, Betsy Ogburn talked about like a collaborative. Is there going to be some sort of formal combining of these results somehow, or is it really just kind of up to whoever's willing to share the data?
Yeah.
I know there's opportunity costs on both sides, like the sharing
Yeah.
versus not sharing in different designs and so forth.
Yeah. I mean, what's happened so far, so phase 1, phase 2 trials, those are being done by the manufacturers and that you know, that they're developers. So that makes sense to me. It's these phase 3 trials that you know, we're advocating that these be combined. That's where the shared resources become very valuable. The NIH, NIAID is also planning a trial. I'm not sure of the details. I know the University of Washington, Fred Hutch is working on that as well. But so I think what we're going to see is a mix of probably 2 pretty large phase 3 trials, these combined adaptive trials. So what the NIAID is planning and what WHO is planning, and then I mean, I wish they would work together. I don't know if that will happen. And then also independent trials from manufacturers. And our goal, you know, the parallelization, you know, is good. We're just trying to prevent where every trial is a small trial, because like what Betsy was talking about, it just becomes hard to interpret the results. It just really complicates the landscape. It's easier if you can keep things simple and make them fast by being big.
Right. I'm going to call on Jay Park, who's had his hand raised for a question, and then we're going to go to Victor Liucci.
Thank you so much, Natalie. I'm a huge fan of your work and as well as your Twitter. I was actually I was one of the collaborator of the group that made the COVID-19 tracker. I think you were aware of it. I think you retweeted our link. I actually have 2 questions. I don't know if I have time for both.
Maybe Jay, just so we have everybody get a chance, you could ask one that is a higher priority, and we can cycle back.
Okay, sure. So,
oops,
We lost you.
that was my fault. Sorry, Jay. Go ahead.
No problem.
Put your hand down, and I accidentally muted you. Sorry about that.
No problem. And so I'll just ask the one question. You know, I think the geospatial analyses to catch the hotspots of the that certainly makes sense, a lot of sense. And, you know, I think our group in support work with the Bill and Melinda Gates Foundation has, you know, supported a lot of these precision public health approaches where you could capture the disease at the right location at the right time. I was wondering if you could elaborate on some of the feasibility challenges of setting up trial sites, you know, not in addition to just training the staff and setting the central randomization system. And
Yeah.
would you have go into more of the manual randomization if the EDC system can't be implemented? And I won't ask into the sort of the cold chain issues that exist with vaccines, because of, I've worked with a lot of in different resource limited countries in Africa, and manual randomization unfortunately is the sometimes only way to go, for an example.
Yeah, we're still, there's always some learning as you go, and there are operational challenges. I would say that what we were able to do in Guinea sort of gives us more confidence, and because they were able to maintain a cold chain through these like really fancy tubes, this technology that allowed you, because that was brought out to sort of these remote sites. There are going to be, so the idea is, the kind of idea where we're moving is to try and get some of the regulatory approvals at a national level, and then maybe have some flexibility about where you go within a country. So, so get things kind of set up in a centralized manner, but then have some level of mobility in terms of where the teams actually go to go out and vaccinate. So you can be a little more nimble in your decision making. The randomization, is funny. I was told that WHO, the randomization is centralized there, and they're under so much attack. Their systems are under so much attack that they've actually had some challenges there, because they have to have these I don't know, firewalls or so there have been some practical challenges that were not expected, and that's come up with the therapeutic trial, but hopefully some of those will be addressed in time for the vaccine trial. So, so yes, those are all challenges, but I think they can be overcome to some degree. So yeah, but it's tricky.
Thank you, Jay. We're going to go now to Victor Liucci. I hope I'm pronouncing your name correctly.
That sounds good. Yeah, I'm at Westminster College in Fulton, Missouri. And my question has to do with your thoughts on vaccines that prevent versus vaccines that reduce severity. I think the Oxford one I had read about seems like it does that.
Yeah, that's a great question. So, so basically, the primary endpoint that we've been talking about for these trials is the prevention of disease, symptomatic disease, or of any severity, but you could focus on, there are secondary endpoints about preventing severe disease. So that's different from preventing infection entirely, because there are some vaccines where people can still be infected, but they just don't manifest severe symptoms. That is obviously not as good, right, as like sterilizing immunity or something, a vaccine that actually prevents infection entirely, because we don't want, then you need to vaccinate way more people to actually have the public health impact, because if people are still spreading, then that's a problem. Yeah, I guess my reaction is that we just have such a great need that we're going to start out with what we can get, and then always hope for something better to come up behind it. So even something that could reduce the severity of symptoms would still have an important impact, certainly at least to the people who are vaccinated. So yeah, but there are questions about kind of what is the lower bound, how much would you accept a vaccine that was only 50% efficacious or 40% efficacious, or you want something higher? Certainly if it's not preventing infection, is it worth that? So those are all considerations that, I'm sure that discussion will play out a lot, especially now that those results have come out. Yeah.
Thank you, Victor. So Natalie, we have a question from Christian Andrade. And Christian, I tried to unmute your microphone. If you'd like to ask the question yourself, you can go ahead. So I'm seeing Christian online, but it doesn't appear that the mic is easily muted. So I'll ask the question. Are trials and data gathering designed to control for the racial disparities that we've seen in early case and death reports?
Yeah, I think a lot of those disparities have to do with exposure and structural reasons why certain, are they designed to, the idea is to include a lot of different people from a lot of different populations. And if you have the mobility, you can go out to populations that may not otherwise be as accessible, that are hotspots, and sort of set up shop there and be able to get a representative population. And the geographical focus, by including a lot of geographical regions, we're going to get, the goal is to try and capture the whole, as much of the world as we can. But it's whether that's adjusted for in the analysis. No, we're just randomizing big numbers. That's the goal, and an individual randomization within locations. So the thought is that would be balanced, and we can certainly analyze a lot of things in the end. But the goal is just sort of on average, does the vaccine work?
Thanks, Natalie, and thanks, Christian. So we have another question from João Alves. And I've unmuted your microphone if you'd like to ask your question.
Okay, thank you. Thank you for the opportunity. My question is that if from the data that we currently have available, is it likely that we would witness significant mutations on this virus that would invalidate or somehow make inefficient any vaccines that we are currently developing, in a way that we would need to develop a new vaccine every year or every season, as we do for other diseases like flu?
Thanks. Yeah, I haven't seen any. I think what I've seen so far is pretty stable. So I wouldn't expect the same challenges. So I'm not a virologist, but but it just, the people I've heard from indicate that it's pretty stable. So that makes things easier.
Okay, thank you.
Thanks.
Okay, so we're at about 12:53. I would like to make just one clarification. Betsy Ogburn reminded me that Natalie is one of the people working on the COVID collaboration platform that she talked about a couple weeks ago. I did know that, and I think I just didn't say it. So anyway, thanks for reminding me of that, Betsy.
Betsy on. Hi, Betsy.
And so I think we're going to bring today's session to a close. I've said this to a few people. I noticed the Zoom protocol has been sometimes run the meetings right to the top of the hour, and then you have another one. I'd like to give people at least a few minutes to get to their next engagement. But Natalie, if you could stand in line for a couple minutes so we could just deal with administrative details.
Sure.
But Natalie, I'd just like to say thanks so much for your work on all this, for your really clear communication on data-related issues in COVID, and for helping us understand what's happening with vaccine research.
Thanks. Yeah. Thanks for having me. That was fun.
Okay, we're going to be taking at least a 1-week hiatus next week. Brown graduation is this weekend, and I think that folks will scatter, but we're going to try and put together 3 to 4 more of these in the month of June. So please keep your eyes open, and thanks everybody for joining.